Welcome to topic 14. Today we're going to have a look at one of the most fascinating and also the least understood organs of the body, the brain. I've split topic 14 into two separate PowerPoints. Part A is going to look at the brain itself. Part B is going to look at the cranial nerves that come directly off of the brain. And in this first part, I'll give you a brief introduction into the brain, and then we'll go through the brain, looking at the different regions and talking about their structure and also what they do. The brain does a whole lot of things, some of which we're consciously aware of, and some of which we're not. And of course, whether or not we're conscious of these things is also a product of the brain. The brain registers sensations, and using those sensations, it brings about a perception of the world that surrounds us. So sensations are correlated with other sensations, and they're compared to stored information that we have so that we can make decisions and take action. The brain is the center for intellect, emotion, behavior, and memory. I quite like this quote by Emerson Pugh. He was a research engineer at IBM for a number of years, and he stated that if the human brain were so simple that we could understand it, we would be so simple that we couldn't. Kind of clever. The brain is an enigma, surrounded by a conundrum and shrouded in mystery. The brain is also remarkably soft. Now, you might not think that if you've done a dissection in the lab, because the brains that we dissect, the sheep brains, are pretty much like rubber. And the reason for that is the fixation process. So what they do with tissues, like the brain, if they're going to preserve them for dissection, is they treat them with chemicals like formaldehyde and ethanol and other alcohols. And what these chemicals do is they cause the proteins to cross-link, to form bonds to each other. That means that the tissue can't be broken down by bacteria, but it also changes the property of the tissue. This is what a fresh brain looks like. This is a fresh human brain. You can see it's very soft. It's actually about the consistency of tofu. You can cut it with a butter knife. It's kind of a soft pink color. Most of that pink color is coming from very fine blood vessels, capillaries within the pia mater. And in this picture here, you can see the dura mater as well on the left. There's a link to this video below that shows a fresh human brain. And I suggest you have a look at that because the brain is perhaps quite a bit different than you might think. It is a lot softer, it's very, very fragile, and it has to be protected by the cranium. Recall that the brain and spinal cord are surrounded by three protective layers known as the meninges. We have the pia mater, which is in direct contact with the nervous tissue, and then we have the arachnoid layer. It's called the arachnoid layer because it's kind of web-like, and arachnoid means spider. Now the arachnoid layer has these tendrils that come off. They're known as trabeculae, and they connect the arachnoid to the pia mater. That space beneath the arachnoid that contains these trabeculae is known as the subarachnoid space, and we have a fluid that moves through that space. That's Cerebrospinal Fluid, or CSF, and it circulates around the brain and spinal cord. Now, outside of the arachnoid mater, we have the dura mater, which is a really, really tough layer. Over top of that, we have the bone of the skull, so the bone that makes up the cranium. On top of that, we have the periosteum. Remember, that's connective tissue that's found on the outside of bones. Above that, we have something known as the aponeurosis. This is a thin sheet of connective tissue to which many of the muscles of the face and head connect. People that can wiggle their scalp, they can contract and pull on that aponeurosis. And above that, we have the skin. Now, also note that within the dura mater, we can have cavities known as sinuses. And you're seeing one outlined in red. I'll just put a little X in the middle here. This is a channel running through the dura mater through the longitudinal fissure that separates the two hemispheres of the brain. and it's going to collect cerebrospinal fluid and move it back into the circulation of the blood. There are cavities known as ventricles within the brain, and these cavities are remnants of the lumen of the neural tube that we see in the embryo. There are two lateral ventricles. There's one in each hemisphere of the cerebrum, and they're connected together through a third ventricle in the middle of the brain. The third ventricle connects to the fourth ventricle. The fourth ventricle is at the bottom of the brain between the brain stem and cerebrum. And the fourth ventricle connects to the central canal that runs the entire length of the spinal cord. In the anterior view on the left, you can get a good idea of how these different cavities connect to each other. Now, one thing to note, just in case you're wondering, if you look at the diagram on the right, we're looking at the brain from the side, and you might be wondering what this structure is. That's something known as the interthalamic adhesion. It's basically a connection between the two parts of the thalamus. What's kind of interesting is 20% of people don't have that connection at all, and some people have two. It's also a bit bigger in women in general. Why? Nobody knows. That cerebrospinal fluid is rather important stuff. The average adult has 80 to 150 mils of this fluid, and we're constantly recycling it and refreshing it. So what does it do? Well, it provides mechanical protection. The brain and the spinal cord are literally floating in this fluid. So it will help soften any impacts that might occur between the nervous tissue and the bones that surround it. It provides chemical protection, and it helps to maintain optimal ionic concentrations within the nervous tissue. So the nervous tissue, of course, contains neurons, and neurons create action potentials by moving large quantities of ions back and forth across their plasma membrane. Those ions are being taken from the cerebrospinal fluid. Also, there's circulation that occurs of this cerebrospinal fluid, and that allows your body to remove waste products from the nervous tissue, and at the same time it nourishes the nervous tissue. It provides it with oxygen and also with nutrients. Cerebrospinal fluid is generated within structures known as choroid plexuses. These are structures that contain a high concentration of rather leaky capillaries. The capillaries leak out plasma, and then that plasma will move through cells known as ependymal cells. We'll talk about those in a moment. That fluid seeps into the ventricles as cerebrospinal fluid, and that fluid is circulated, as you can see, through the ventricles of the brain, around the exterior of the brain, around the exterior of the spinal cord, and through the central canal of the spinal cord. And you can see in diagram B here that we have these choroid plexuses in each of the ventricles of the brain. If you had a chance to dissect the sheep brain, you would have seen these as rather dark brown structures. The outer surface of the choroid plexus and also the entire lining of the ventricle is made up of a continuous layer of ependymal cells. The ependymal cells are held together by tight junctions within the choroid plexus, but they're not elsewhere within the ventricle. So let's take a look at the choroid plexus. We have lots of capillaries within the choroid plexus. It's very heavily vascularized, and those capillaries are very leaky. They're fenestrated. Remember, that means they contain windows or holes. And those holes are going to allow plasma to seep out, and that's going to include big proteins and other large molecules. That material can't get directly into the ventricle, though. It still has to pass through the layer of ependymal cells. So the ependymal cells, as I mentioned, are held together by tight junctions within the choroid plexus. That means that fluid can't seep between the cells. They're sealed together. So anything leaving the blood on its way to the cerebrospinal fluid of the ventricle has to pass through an ependymal cell. The ependymal cell has to actively take it in and then actively transfer it to the other side, or it has to have channels that allow for facilitated movement of these materials. So ultimately, the ependymal cells will determine what gets into the ventricle. So we have water and we have ions and vitamins and oxygen, etc. And that's what's going to constitute the cerebrospinal fluid. This is how that fluid is made and maintained. At the same time, waste products are going to be going in the opposite direction. They're going to be taken up into the capillaries and then eventually transported to the kidneys to be disposed of. Now, within the nervous tissue, we have capillaries as well. So the cells aren't getting all of their nourishment and oxygen from the cerebrospinal fluid. They do get some from embedded capillaries as well. But note that the capillaries are surrounded by astrocytes. Astrocytes are glial cells. Those are support cells. And note that they have extensions, kind of like appendages that come off and wrap around the entire surface of the capillary. So this provides another barrier known as the blood-brain barrier. Anything within the capillaries has to pass through the astrocyte before it gets to the nervous tissue. So the astrocytes are going to determine what leaves the blood and comes into contact with the rather delicate and sensitive neurons. Here you're seeing a sagittal section of the brain, and again we have these 4 choroid plexuses, so one within each of the 4 ventricles. They're producing this cerebrospinal fluid that circulates around the brain. Ultimately, it's going to be recollected by structures that are known as arachnoid granulations. They kind of look like little cauliflower heads. They are going to extend into the superior sagittal sinus, which runs between the 2 hemispheres of the brain. That fluid is recollected and ultimately taken back to the circulatory system. Blood makes its way to the brain through the carotid arteries and the vertebral arteries. It makes its way to the choroid plexuses, where the plasma is going to seep out. It's going to be filtered by the ependymal cells and give rise to cerebrospinal fluid. So we have 1 choroid plexus in the 4th ventricle, 1 in the 3rd, and 1 in each of the lateral ventricles. That cerebrospinal fluid is going to move through the ventricles, make its way into the subarachnoid space, and then be collected by arachnoid granulations, also known as arachnoid villi. That fluid is then dumped into the sagittal sinus, and that makes its way back to the heart through the venous system. These arachnoid villi are extensions of the arachnoid mater. They project through the dura and into that superior sagittal sinus. And this allows us to reabsorb about 20 mils of cerebrospinal fluid every hour. In addition to the superior sagittal sinus, we have other sinuses that collect used cerebrospinal fluid within the cranium. They're all embedded within folds of the dura mater known as fulxes. We have one fold that separates the 2 hemispheres of the cerebrum, and we have another fold that separates the cerebrum from the cerebellum. A sinus is essentially a swollen vein, and these structures are going to lead blood back to the jugular vein, and then it can make its way back to the heart. Alrighty then, now that we've looked at an overview of the brain and we've talked about cerebrospinal fluid and how it's circulated, we'll start looking at the different regions of the brain and talk a bit about what they do. What you're seeing here are images that were generated by computer tomography, CT scans or CAT scans. So on the right, we have a frontal or coronal section. You can see it passes through the brainstem. On the left, you're looking at a series of sections. Remember that CT scans are essentially X-ray scans, but we're creating X-ray images within a thin plane. And then we can put those together to build a 3-dimensional image, or, like you're seeing here, a really neat little movie moving down through the brain looking at transverse sections. And you can see the ventricles quite clearly as we go through the brain. The brain and spinal cord develop from a neural tube, as we talked about extensively in our previous topic. Remember that the front part or anterior part of that tube is going to expand to give rise to the brain. On the left is a diagram of the early embryonic brain. The neural tube has swollen to create three distinct regions. We have the forebrain, the midbrain, and the hindbrain. And we can continue to talk about the brain as consisting of these three distinct regions in the adult as well, and that's what you're seeing on the right. Notice that the forebrain has these buds or out pockets coming off that will give rise to the eye and also to the optic nerve. The optic nerve is unusual among the cranial nerves and nerves in general because it has a hollow in the middle, and it has that hollow because it's an offshoot of the neural tube. These three regions are already established in the 3 to 4 week embryo. The forebrain is going to divide further into two parts. It divides into the telencephalon, which is going to give rise to the cerebrum, and the diencephalon that gives rise to the thalamus, hypothalamus, and epithalamus. Notice again we have these eye cups. Again, they give rise to the optic nerve and also to the retina at the back of the eye. The midbrain doesn't divide any further, but the hindbrain divides into two parts. The metencephalon, which is going to give rise to the pons and the cerebellum, and the myelencephalon, which will give rise to the medulla oblongata. In summary, we have two broad divisions to the nervous system. We have the central nervous system and we have the peripheral nervous system. The central nervous system is further divided into the brain, or encephalon, and the spinal cord. The encephalon can be divided into the forebrain, midbrain, and hindbrain. The forebrain consists of the cerebral hemispheres and the diencephalon, which is made up of the thalamus and hypothalamus. The hindbrain is developed into the metencephalon and myelencephalon. The metencephalon is the cerebellum and pons, and the myelencephalon is usually just referred to as the medulla or medulla oblongata. The peripheral nervous system consists of two parts. The somatic system, which consists of voluntary nerves or skeletal nerves, and the autonomic system, which is divided into two divisions, the sympathetic division and the parasympathetic division. And we'll come back and talk about the autonomic system in our next topic. Now we'll go through and look at the bits and pieces that make up these regions, and we'll start at the bottom with the brainstem. The brainstem connects to the spinal cord and it supports the cerebrum and the cerebellum. It consists of the medulla oblongata, the pons, and also the midbrain. The medulla oblongata is a continuation of the spinal cord. It's essentially a swelling at the top of the spinal cord. And because of that, it's a highway for information. So we have sensory tracts, bundles of axons that are coming up from sensory structures throughout the body, making their way through the medulla up to the higher areas of the brain. And likewise, we have motor tracts that are coming from higher areas of the brain and passing through the medulla oblongata on their way to effectors throughout the body. We have 5 cranial nerves that originate from nuclei within the medulla oblongata. Remember, a nucleus is a collection of neuronal cell bodies. Their axons leave together and form a nerve. There's a lot of very fundamental, important control that goes on within the medulla oblongata. For instance, we have cardiovascular centers that control the force and rate of heartbeat, and also control the vasodilation and vasoconstriction of blood vessels. So it regulates blood pressure. There's a respiratory center. We have this rhythmicity area that sets the basic rhythm of breathing. We also have information coming out of the cerebellum or entering the cerebellum that has to pass through the medulla. And finally, we have a lot of important reflex centers within the medulla oblongata. Coughing, sneezing, gagging, and swallowing, the aspects of these processes that you don't really have any control over, are coordinated by the medulla. So we have those 5 cranial nerves, nerves 8 through 12, and then we also have sensory nuclei that are present within the medulla that process the sense of touch, so they're receiving inputs, and also process proprioception from the lower body. Remember, proprioception is the perception of where your limbs are with respect to your body and how your body is positioned in 3-dimensional space. The medulla contains the nuclei for the vagus nerve, cranial nerve number 10. And remember how important the vagus nerve is. It travels down to the chest and into the abdomen and controls breathing and heart rate and aspects of digestion. We have sensory inputs from the carotid sinuses and from the aorta that carry information about carbon dioxide levels, oxygen levels, blood chemistry, blood pH, blood pressure. Because the respiratory and cardiovascular centers are found within the medulla, damage to the medulla is a very serious thing. So disrupting those centers can very quickly lead to death. Non-fatal injury can cause paralysis, loss of sensation, but also irregularities in breathing and heart rate that can be very serious. The pons is a swelling of the brainstem that's located immediately superior to the medulla. It communicates with the spinal cord and other parts of the brain by way of tracts. And I'm just realizing now I haven't really clarified what a tract is. A tract is a bundle of axons that makes up part of the central nervous system. Quite often, a tract is going to be surrounded by other nerve tissue. Now, that's not the case with a nerve. A nerve is an isolated bundle of axons, typically surrounded by its own layers of connective tissue. And a nerve makes up part of the peripheral nervous system. So what does the pons do? Well, like the medulla oblongata, it plays some very important homeostatic roles. It's involved in maintaining respiration and controlling respiration along with the medulla. It's also going to relay nerve impulses that are involved in skeletal movements, voluntary movements, from the cerebral cortex to the cerebellum. And we'll talk about what the cerebellum does in just a moment. Additionally, it gives rise to cranial nerves 5 through 7. The midbrain is the superiormost portion of the brainstem. It's a rather small area, but there's a lot of interesting stuff happening here. It receives visual inputs before they're passed onto the visual cortex of the occipital lobe. So there's a bit of pre-processing that occurs here. It's responsible for coordinating eye movements with visual stimuli. So if you see something moving in your peripheral field of view, you automatically, as a reflex, turn your head and your eyes towards it. And that's an activity that's coordinated in this region. Also, as you move your head, and let's say you're staring at something, your eyes have to move relative to your head in order for you to continue to be staring at that object. That's another sort of movement that's coordinated in this area. A similar thing happens with auditory stimuli, so sounds. The information that comes from your ears is also processed here first before it goes on to the auditory cortex. Again, this region is going to help coordinate movements of your head with these stimuli. If you hear a loud noise, you immediately turn towards it You don't have to think about it, and your brain figures out where the sound is coming from because, of course, the sound is going to be louder in the ear that's facing the sound more directly. Here we see a transverse section of the midbrain. The tectum is responsible for coordinating reflex reactions to auditory and visual stimuli. The geniculate nucleus receives visual input. And note that the ocular motor nerve and also the trochlear nerve originate from this region of the brain. That makes sense, of course, because the ocular motor nerve is the major nerve innervating the extraocular and intraocular muscles of the eye. Also within this region we have two areas known as the red nuclei, and they're involved in basic motor coordination that occurs during walking. Specifically, they're responsible for you swinging your arms when you walk in a coordinated fashion that helps you maintain your gait and your balance. Finally, we have the cerebral peduncles. These are the connections with the cerebrum. The diencephalon sits atop the midbrain. You can see it's composed of a thalamus, which has a right and left lobe to it, and under that we've got the smaller hypothalamus. Hypo meaning below, so below the thalamus. The diencephalon is the blue part of this diagram, and you can see it kind of looks like the head of a seahorse. Although we're going to talk about a structure in a bit that looks a lot more like a seahorse. Underneath the hypothalamus, we have something called the hypophysis, although you probably know it as the pituitary gland. In this coronal or frontal section of the brain, you can see that the hypothalamus and the thalamus surround the third ventricle. The thalamus is one of the primary relay centers within the brain. Sensations that come into the brain go to the thalamus first, and then the thalamus sends off signals to the cortex, the surface part of the cerebrum. And that's where interpretation occurs. So that's where you're going to consciously interpret the information you're getting from the thalamus and decide what you're going to do about it. Now there's one exception, and that is smell. Smell does not travel through the thalamus. Those sensations are carried to the limbic system, which we'll talk about later. The thalamus can also register some conscious recognition of pain directly, and temperature, and awareness of light touch and pressure. The hypothalamus is a major regulator of homeostasis, and we talked about this when we talked about the endocrine system. Remember that it produces hormones. The hormones travel to the pituitary gland, where they may bring about the release of additional hormones. It controls the autonomic nervous system, which is the portion of the nervous system that regulates involuntary homeostatic responses. It regulates emotional and behavioral patterns. It's involved in eating, drinking, satiety, which is fulfillment of hunger, and thirst. It monitors and regulates body temperature. This is where the thermostat for your body is located. And it also regulates circadian rhythms, so day-to-day rhythms and states of consciousness. So it's involved in your sleep cycle. The hypothalamus contains a number of discrete nuclei that have discrete functions. And we won't get into the names of all these, but just realize that some of them consist of the cell bodies of neurosecretory cells that produce hormones. Some of them are areas that monitor temperature. Some of them are areas that regulate temperature and bring about sweating and shivering. So there's a whole lot going on here. This area is also receiving somatic and visceral inputs. Taste, smell, and hearing is to some extent being processed here. And some areas, particularly the mammillary region, is involved in recollective memory. Before we move on to the epithalamus, I just want to draw your attention to the interthalamic adhesion, also known as the intermediate mass of the thalamus. This is a physical connection between the right and left parts of the thalamus, but it doesn't appear to be essential. I mentioned before that it's missing in 20% of people. It doesn't seem to cause them any problems. Anyway, on to the epithalamus. The epithalamus is a little nub of tissue that lies superior and posterior to the pineal gland. The pineal gland secretes melatonin. Together with the activities of the hypothalamus, melatonin is going to regulate circadian cycles. There's one component of the hindbrain that we haven't talked about yet, and that would be the cerebellum. Cerebellum literally means little brain, and it kind of looks like a separate little brain hanging below your cerebrum. It's divided into two hemispheres, and it's divided by a raised structure, a ridge, known as the vermis. Vermis means worm. The cerebellum is responsible for correcting voluntary muscle contractions, movements, and postures that might otherwise cause you to fall down. So let's say you're walking forward. Now all you have to think in your head consciously is that I need to put one foot in front of the other. But of course the process of walking is far more complicated than that. A number of different muscles have to contract, and they have to do that in a very specific sequence. If you move a leg forward, that throws you off balance. But fortunately, your cerebellum is compensating for your lack of understanding about what's actually going on. So your cerebellum is coordinating all these different activities and ensuring that you stay upright. It's also receiving sensory inputs that are proprioceptive, that tell you whether your limbs are bent or straight, and whether you're standing up or lying down. It's involved in your sense of equilibrium and your sense of balance. The outer gray matter of the cerebellum is known as the cerebellar cortex, and it's thrown into folds and ridges that are known as folia. Within the middle of the cerebellum, as you can see in this mid-sagittal section, we have white matter, and that consists of tracts that carry signals across the cerebellum and also into and out of the cerebellum. This white structure is referred to as the arbor vitae, which means tree of life. The remainder of this topic is going to be spent discussing the largest portion of the human brain, the cerebrum. The surface layer, the outer 2 to 4 millimeters, is comprised of gray matter, and this makes up what's called the cerebral cortex. The cortex contains the cell bodies of billions of neurons. The white matter that makes up the interior of the cerebrum is comprised of axons. The cortex is thrown into these small folds that are known as gyri, or if we're talking about a single fold, it would be called a gyrus. We also have shallow grooves that are known as sulci. A single one would be a sulcus. But we also have very deep grooves that are known as fissures. We can see these structures a bit more clearly on these diagrams. Again, the cerebral cortex is the outermost layer of gray matter. It contains cell bodies. The white matter, or subcortex, contains axons. Each one of the little folds that you can see in the coronal section is a gyrus. Each one of the shallow grooves is a sulcus. And then we have these very, very deep grooves that are known as fissures. So we have the longitudinal fissure here along the back of the brain in the mid-sagittal plane that divides the cerebrum into two hemispheres. There's a couple more fissures here that are obvious as well. So we have a fissure here and a fissure here, and these are known as lateral fissures. The tracts that make up the white matter of the cerebrum connect different parts of the cortex to each other. They also connect the cortex with the rest of the brain and ultimately with the spinal cord as well. The cerebrum is divided into two hemispheres, the right and left hemisphere, by the longitudinal fissure, but that fissure doesn't completely cut the cerebrum in half. The two halves are still connected through a structure known as the corpus callosum. The corpus callosum contains white fibers, axons, that can run transversely across the cerebrum. Each hemisphere is divided into 4 lobes, and those lobes are named after the bones that they underlie. So we have the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. In this model, you're seeing a representation of the direction that the different axons and tracts travel in. But just note that the white portion of the cerebrum consists of a highway of fibers. We have fibers that are running from one area of the cortex to another. We have fibers that are running towards the corpus callosum and then across to the other hemisphere. And then we have other fibers that are running to other parts of the brain and down into the spinal cord. The corpus callosum is a structural connection between the two hemispheres. But it's a lot more than that. It is white matter, and it consists of 200 to 250 million axons. And these axons are carrying information from neuronal bodies that are contained within the cortex. So information is coming from the cortex on the left hemisphere to the right, and vice versa. Let's talk about some of the general features of the cerebrum. Again, I've talked about this a few times and I'll talk about it a few times more, but I just want to really drive this into your brain, so to speak. A gyrus is an elevated ridge on the surface of the cerebrum. A sulcus is a shallow groove that separates adjacent gyri. An important sulcus is the central sulcus because it divides the frontal lobe from the parietal lobe. So this is kind of a landmark sulcus. And this sulcus is found right here. And we'll come back to that. A fissure is a very deep groove. It's going to divide the brain into large regions or lobes. And we have a few important fissures to talk about. One of them we've already mentioned is the longitudinal fissure. You can't see it in this diagram because it runs down the mid-sagittal plane of the brain and divides the cerebrum into 2 cerebral hemispheres, the left and right hemisphere. Next we have the transverse fissure, which separates the cerebrum from the cerebellum, and that would be found here. So we have the cerebellum at the bottom there, and of course the cerebrum above that. We also have the Sylvian or lateral fissure, which divides the temporal lobe from the frontal and parietal lobes. And that would be found here. You can see this in a bit more detail here. Now, one thing I just want to draw your attention to, because it might confuse you a bit, is the fact that the lateral fissure or Sylvian fissure is very commonly referred to as the lateral sulcus or Sylvian sulcus. In fact, it's more commonly called a sulcus, even though it's actually a fissure. In fact, if you look up the definition of the lateral sulcus, it will tell you it is a deep fissure that separates the frontal lobe and parietal lobe from the temporal lobe. Just something that's kind of weird. There's a lot of weird stuff when it comes to medical terminology. Well, how do we define the different major regions of the brain? Sometimes it's obvious, sometimes not so much. First of all, we have the transverse fissure, which separates the cerebellum from the cerebrum. That's pretty obvious and pretty straightforward. Keep in mind that we would have a membrane composed of dura mater that would sit within that fissure. If we're looking at the cerebrum, we have the longitudinal fissure that's going to separate the cerebrum into the left and right hemispheres. Now, looking at one of the hemispheres, we're going to have a sylvian or lateral fissure that will separate the temporal lobe from the frontal lobe and also from the parietal lobe. The frontal lobe is going to be separated from the parietal lobe by the central sulcus. That's not quite as obvious because, of course, a sulcus is not nearly as deep as a fissure. And I should point out that if you look at some diagrams on the web, it may not be quite so clear. The occipital lobe, as you see in this diagram, isn't clearly separated from the other lobes of the brain. Instead, it's isolated by the fact that it lies under the occipital bone of the skull. So here we're looking at the boundaries between the bones of the cranium instead of clearly defined regions. Okay, so we have 4 lobes of the cerebrum that are clearly recognizable, and they do different things. We have the frontal lobe, we have the parietal lobe, we have the occipital lobe, and we have the temporal lobe. But depending on who you talk to, there might also be a 5th lobe, and that's known as the insula. The insula is a deep lobe. You have to pry apart the temporal lobe from the frontal lobe and the parietal lobe in order to see it. To expose the insula, you have to take that lateral fissure and spread it wide open. And that's what's being shown here on the left. The green is the insula. If we look on the right-hand side of this slide, you can see at the top, we've got a transverse or cross-section of the brain. The insula would be this portion of the exposed cortex here and over here. And down the bottom, we have a frontal or coronal section. The insula would be this surface here and here. And of course, the lateral sulcus or lateral fissure would be here and here on the top diagram, and here on the bottom diagram. So before I go forward and start talking about the different functions of the different lobes and regions of the cerebrum, I just want to make it clear that it's actually not that clear. So if you look online, you'll see that the frontal lobe does this and the parietal lobe does that, and it seems pretty straightforward. But unfortunately, things are a bit messier than that. So although the different lobes do have specializations, we have redundancy occurring throughout the brain. The frontal lobe, for instance, is involved in intelligence and reasoning and behavior, but those processes are covered to some extent by other parts of the brain. The parietal lobe, for instance, is also playing a role in intelligence and reasoning. The temporal lobe is usually associated with language, but some of the language processing that occurs within our brain is also covered by the parietal lobe. The brain is also somewhat malleable and plastic. If you damage one part of the brain, especially if you're a younger individual, that processing, hopefully, can be taken over by another part of the brain. Another really common myth is that you only use 10% of your brain. And I imagine that comes from the idea that the cortex contains the cell bodies of neurons. But of course, the interior of the brain is also really important. It conducts signals. So the idea that you're only using 10% of your brain is bull feathers. Anyway, we're going to look at the predominant functions of the different parts of the brain, but I just want you to realize that it's not quite that simple. There are many functions. Memory is a really good example that occur throughout the cerebrum. All right, so keeping in mind that we have overlap between the different regions of the cerebrum, let's take a look at the specifications. We'll start with the frontal lobe. The frontal lobe is associated with memory formation, as is much of the cerebrum. It's associated with emotions, and more specifically, it's associated with decision making, reasoning, and personality. Personality is specifically associated with what's termed the prefrontal lobe. The prefrontal lobe would be this area in here. With respect to function, we can recognize a few primary areas within the frontal lobe. Remember that we have the central sulcus that's going to separate the frontal lobe from the parietal lobe, and this serves as an important landmark. Anterior to the central sulcus, we have an area known as the primary motor cortex, or precentral gyrus. And that would be right here. Precentral, referring to the fact that it's ahead of or in front of the central sulcus. Of course, the gyrus refers to this fold that is just ahead of or anterior to that sulcus. And we'll talk about the role that it plays in a moment. We also have Broca's area, which is involved in language, and this is found just anterior to that motor cortex and above or superior to the temporal lobe. We also have the orbitofrontal cortex, which is found down here. And this is involved in personality and behavior. Underneath that, of course, we have the olfactory bulb, which is going to be collecting inputs from olfactory receptors within the nose. So it's involved in the sensation of smell. The primary motor cortex, or precentral gyrus, is the fold that exists immediately anterior to the central sulcus. It controls voluntary movements of the body. Broca's area is going to control facial movements that are involved with speech, and it's also involved in language comprehension, as we'll see. Usually, this area is focused on the left frontal lobe, although it can be on the right frontal lobe, and its position is a little bit variable. If you have damage to this area and you're fortunate, it can actually move slightly. We can have adjacent areas of the brain that take over this function. The orbitofrontal cortex is involved in decision making, reward, punishment, associations, taste, sensory appreciation, emotion, and behavior. A lot of really critical stuff going on there that we think of as the kind of stuff that makes us human. The punishment associations are kind of interesting. If you're on a diet, let's say you have a sweet tooth and you really appreciate things that taste sweet. You're on a diet and you stop eating candy bars. The reward punishment associations that are occurring during that diet are taking place here. So you feel really bad, really guilty because you cheated on your diet. You have this area of your brain to blame. The olfactory bulb, which hangs underneath that, is the extension at the end of cranial nerve 1. And this is going to receive sensory inputs from the sensory receptors, the olfactory receptors within the nose. So the olfactory bulb is involved in smell. If you're a biologist and you're trying to figure out what a certain structure does, one strategy that you might employ is to simply cut that bit out and see what goes wrong. Now, you can't generally do that with humans, nor should you. Instead, you can collect information from people that have had injuries. Unfortunately, this is how we know what we know about the brain. A lot of it comes from people that have had traumatic injuries that have damaged part of their brain, and we have documented what went wrong as a result. A really good historic example of this is the case of Phineas Gage. Now, apart from having a really cool name, this guy suffered from a rather terrible accident. He was a railroad foreman in Vermont, and what he was doing is he was packing down explosives, tamping them down into rock using a big metal rod. The explosives went off, and that metal rod was propelled up through his face and out the top of his skull, as you can see in the diagrams here. As it traveled through his skull, it destroyed a large portion of his frontal lobe. Remarkably, he didn't die. The doctor that was attending to him pulled this rod out, and apparently a big chunk of this guy's brain fell out into the doctor's hand. But he didn't bleed to death. He lived for another 12 years. But he wasn't the same person. He went from being a very quiet, reserved, well-mannered gentleman to being someone that was rather outspoken, someone that was swearing at people, someone that was even urinating on the street. He became a totally different person. And this was the first really well-documented case of injury causing a change in personality. This is a case where we saw for the first time that the frontal lobe performed a very specific function. And again, this kind of injury has given us a lot of insight as to how different parts of the brain are specialized. In the 1940s and 50s, there was an unfortunate procedure that gained a lot of favor, especially within the US. This is something known as the prefrontal lobotomy, and it was performed on people that were institutionalized with severe behavioral disorders. So what they would do is they would take a metal probe, they would insert that through the top of the orbit up into the white matter of the frontal lobe, and they would scramble that tissue. So what that did is it destroyed connections between parts of the cortex of the lobe. In some cases, it proved to be rather helpful in that it prevented reoccurring thoughts and reoccurring behaviors. But in most people, it caused sort of a zombification. It destroyed their personality. So fortunately, this is no longer performed. It hasn't been performed for quite a long time. Now, as you can see in these images that I've taken from the American Journal of Psychiatry from this time period, maybe just some makeup and a new hairstyle might have given you the same results. And I don't mean to be glib with that. I just mean that it's quite remarkable that in this journal article, they're presenting these images of this person, and we're meant to read their mental state. And obviously, they've just presented these before and after pictures where they've just made up this individual. The parietal lobe underlies the parietal bone of the cranium, and its main function is perception and the integration of sensations. It's involved in spatial awareness and also proprioception, which is the awareness of where your body is in three-dimensional space and where your body parts, your limbs, for example, are in relation to each other. Within the parietal lobe, we have the primary somatosensory cortex, which is immediately posterior to the central sulcus, and for that reason it's also known as the postcentral gyrus. We have the somatosensory association cortex just behind that that's going to receive inputs from that postcentral gyrus. And we also have the primary gustatory cortex, which is involved in the perception of taste. The Primary somatosensory cortex, or postcentral gyrus, is receiving tactile and proprioceptive information. It's passing that on to the somatosensory association cortex, where that information is going to be integrated and interpreted. And then once again, we have that primary gustatory cortex, which is involved with the sensation of taste. Unlike the other lobes of the cerebrum, the occipital lobe has a very clearly defined function. It's involved in processing, integrating, and interpreting visual stimuli. At the very back, or posterior, of the occipital lobe, we have an area known as the primary visual cortex. And this is the area where visual inputs first arrive at the occipital lobe. Just anterior to that, we have the visual association area. And this is an area that's going to process that raw data that comes into the primary visual cortex. So again, we have our primary visual cortex, which receives this raw data related to size and color and shape and so on of the object you're viewing. And then you have the visual association area that's going to interpret that information and start to make sense of it. So imagine that you're walking across the road and you see a car coming towards you. You're receiving lots of information at the primary visual cortex, the color of that car, the size of that car. And as the car drives towards you, of course, the apparent size of that car is going to increase. Now, all of that information is going to be sent to the visual association area, and it's going to make sense of that. It's going to tell you that, okay, the size of the car is increasing. That means it's getting closer to me. That information goes to the cortex of the frontal lobe, where you get to make a decision about what to do. Maybe you should get off the road. The optic nerves receive inputs directly from the retina at the back of the eye. If there's an object in your left visual field, that means it's off to the left-hand side, it's going to stimulate the right-hand side of the retina in both of the eyes. That information travels to the right visual area. If you're looking at something that's in your right visual field, so off to the right-hand side, that's going to stimulate the left half of the retina in each of the eyeballs, and that information is carried to the visual area of the left hemisphere. The crossing over of these signals occurs within the optic chiasm. So note that within the optic chiasm, half of the information is going to cross over to the opposite hemisphere of the brain. Now let's take a look at the temporal lobes. They're located under the temporal bones of the skull, of course. They're involved in hearing and also the organization and comprehension of language. They also play a big role in memory formation and retrieval. We're going to take a look at some of the specialized regions within the temporal lobe, and we'll start with the primary auditory cortex. This is receiving inputs from the ear, inputs related to hearing, not to equilibrium. Behind that, we have Wernicke's area. Wernicke's area, as we'll see, is very important when it comes to language comprehension. We also have the primary olfactory cortex. This isn't right at the surface, unlike these other areas. It's deep and anterior to the primary auditory cortex. Recall that most of the special senses come through the thalamus, and the thalamus relays this information. Well, the primary olfactory cortex is receiving information directly from the olfactory nerve. And the olfactory nerve is also passing information to the limbic system. To recap, the primary auditory cortex is responsible for hearing. The primary olfactory cortex is responsible for the sense of smell. This area is not found at the surface, though. It's deep to the surface. Wernicke's area is responsible for language comprehension, and usually this consists of one area that's found on the left temporal lobe only. But that's not always the case. It can be found on the right in some instances. Also note that it's something that's a bit variable between people. So in some people, it might not be exactly where we find it on the diagrams that I'm showing you. Now, if something goes wrong with Wernicke's area, as you might expect, you're generally going to get a problem with language comprehension. People that have damage to this area through a stroke, for instance, are able to form words and sounds, and they're able to rattle off sentences, but the sentences don't really make sense. They've got all the right words, but they've put them in the wrong order. This is sometimes referred to as word salad. All the words are just thrown together. The impairment of language is a condition known as aphasia, and we could be talking about impairments of language comprehension or impairments of language production. If you have damage to Wernicke's area, you have a condition known as Wernicke's aphasia, also known as fluent aphasia. It's called fluent aphasia because people with this condition can form words and they can put the words together into something that sounds like a sentence, but it's not a sentence. The words are jumbled. The sentence doesn't make any sense. What you're seeing in the diagram here is what would happen during a normal conversation. You're listening to someone and you get auditory inputs from the primary auditory area that are passed to Wernicke's area. Wernicke's area is going to interpret that, and also it's going to form a response. So let's say you're going to respond to this person. You're going to take words and put them together to form a sentence. That happens in Wernicke's area, and that information is sent to another area known as Broca's area. And Broca's area is within the frontal lobe of the brain. Within Broca's area, what's going to happen is your brain is going to figure out how to turn that information into sounds. Basically, Broca's area is going to look at those words and figure out what motions of the lips and tongue and larynx are needed to form the sentence. That information goes to the motor area. And then from the motor area, signals go out to the lips and throat and tongue. Now, if you get a jumbled sentence from Wernicke's area, that information is going to be sent ultimately to the motor area, and we're going to get words and so on, but of course they're going to be jumbled. If Broca's area is damaged, again, maybe because of a stroke, but Wernicke's area is working, then you understand how to construct sentences and you know what you want to say, but you have a great deal of difficulty saying it. And that's because Broca's area is not sending signals out to the motor area that will bring about speech. Even though you know what you want to say, you can't form the words, which, as you can imagine, would be quite frustrating. The bundle of axons, or tract, that connect Wernicke's area to Broca's area is known as the arcuate fasciculus. Damage to this results in conduction aphasia, and it's exactly what it sounds like. The brain has a difficult time conducting signals from Wernicke's area to Broca's area. People that have this condition have a very difficult time repeating back something that's said to them. They may lose their way in their own sentences. If you ask them to recite the alphabet, for instance, they may lose track of where they are and what they're doing. There are a number of really good videos on YouTube that show these different types of aphasia, and I've put links up to a few of them. Please take the time to have a look at them because they'll make it a lot clearer and they'll hopefully let you see what these people are struggling with. Let's just take a moment to quickly review some of the areas that we've looked at. We'll look at the sensory areas first. Remember, a sensory area is going to collect sensory information and also interpret those sensory impulses. We have the primary somatosensory area, and that's not shown on here, but that would be right here. So that's the post-central gyrus. We have the primary visual area, and this is at the back, the posterior, of the occipital lobe. And just anterior to that, we would have the visual association area, which provides further interpretation of those visual cues. We have the primary auditory area that's responsible for hearing, and we have the primary gustatory area that's responsible for taste. We've also talked about some important motor areas. So Broca's area is important when it comes to speech, and it communicates with the primary motor cortex. And we'll talk about the primary motor cortex in a bit more detail now. The primary motor cortex is responsible for voluntary movements of the body. It's responsible for contractions of skeletal muscle. Remember, it's that single gyrus that's found just in front of the central sulcus. So it's the pre-central gyrus. The diagram on the right shows that we have distinct areas along that gyrus that control very specific parts of the body. We have larger areas and more neurons dedicated to things like the hands and the lips and the tongue, things that we need to move in very precise motions. The precentral gyrus is responsible for motor outputs. The post-central gyrus is responsible for sensory inputs from your body. What you're seeing here are sculptures, and the sculpture is known as a homunculus, that show how your brain sees your body. On the left, we have how your brain sees sensory inputs. So what we're looking at here basically are the number of neurons within that post-sensory gyrus. You can see there's a lot that are devoted to the hands. You have a lot of very precise, very fine sensory inputs in your hands. You can see that there's a lot of sensory inputs that come from the mouth and also from the genitalia as well. Now compare that to the motor outputs. A large portion of that precentral gyrus is devoted to the hands. We can do pretty amazing things with our hands. We have lots of fine motor movement in the hands. We have lots of motor output to the lips and also to the throat because we require a lot of complex, precise movements of the throat and tongue to bring about language and speech. This diagram shows you some of the association areas of the brain. So again, the visual cortex is composed of two parts. We've got our primary visual cortex, which is receiving information from the retinas of the eye, but then surrounding that, we've got the visual association area, which is taking that information and performing integrative functions. It's looking for patterns, and it's comparing that information to stored information, information that's stored as memories. Within the sensory cortex, we have our post-central gyrus that's receiving sensory inputs, but then behind that, we have this somatic sensory association area that's starting to interpret those inputs. The motor cortex consists of that pre-central gyrus, and that's where motor outputs are going to leave the brain. But ahead of that, we have the somatic motor association area where your brain is starting to formulate these movements. Your brain is saying stuff like, I want to walk over there, and then that information is going to be sorted into the individual movements of muscles that have to occur. That information can move on to that precentral gyrus, and that information can go out to individual muscles. We have the auditory cortex, which consists of the primary auditory cortex, which is receiving signals from the ears, and then we have the auditory association area, which would include Wernicke's area. This diagram here shows you some of the areas of the brain that have been mapped by function. And the numbers aren't random. These are universally agreed upon labels for the brain. So if you were to talk to a neuroscientist about, I don't know, area 21, they would know exactly what you were referring to. Note that this diagram is flipped around from the other diagrams I've shown you, so the anterior of the brain is towards the right. Now, I don't expect you to remember any of these numbers, and you certainly don't have to remember all the parts that are shown here. I've included a few of the more important and more interesting ones on the left. Note that the premotor area, which is number 6 on this diagram, is also referred to as the somatic motor association area. That's how it was labeled in the last slide. And within this area, one of the interesting things that occurs is that we have learned skilled movements that are remembered here. So what does it take to type? What does it take to cross-country ski? What sort of movements do you need to do? So these are things that may have taken you a while to learn, and you've stored that information in this association area so that it can be readily passed to the primary motor cortex. Finally, we have the limbic system, which is a very deep structure in the brain. It's sometimes referred to as the emotional brain or the paleomammalian cortex. Paleo means old. This is an ancient, primordial structure. It's responsible for emotions, especially kind of base primordial emotions. It's involved in long-term memory. It's involved in motivation. And it also plays an important role in olfaction. It's connected to your sense of smell. And this is why your sense of smell can bring about very powerful emotions in some cases, and also is typically linked to old memories. You smell something as an adult, and that might bring back very sudden and powerful memories of your childhood. The inferior portion of the limbic system forms a structure called the hippocampus. Hippocampus is Latin for seahorse. I told you we'd come back to seahorses. This structure is involved in long-term memory, and it also helps with your sense of direction. If you're walking in the woods and you turn left and turn right and turn left, maybe, if your hippocampus is doing its thing, you might know what direction you're facing after all that. Also, interestingly, it's involved in overcoming urges. It helps you overcome the primal urges that are being created by the rest of the limbic system. It prevents you from doing things that you probably shouldn't do. And finally, we have our terminology. There's a lot of it because the brain is a rather complex thing. As I mentioned, we're going to move on to the cranial nerves next. So I will see you there.